Hello, everybody, for our next installment of Journal Club and papers related to aging and longevity. I'm Oliver Medvedik. With us is Steve Hill, Victor Bjork, Michael Lustgarten, and Michael will be chiming in periodically since he knows a heck of a lot more about microbiomes than I do. Put you on the spot. Anyway, the paper we're going to be discussing is a bit different from what we've discussed before. We're going to be looking at the microbiome and how it affects aging, particularly in mammals, specifically in rodents, and how this may impact us and the aging process, and particularly the effects of the microbiome on the health of the gut and inflammation in general. So with that, let me pull up the paper. Share screen. I'm slow with this. That's the right one. I think that's the right paper. Okay. There we go. So, if I got the right paper, you should be seeing Cell Host Microbe 2017, Age-Associated Microbial Dysbiosis Promotes Intestinal Permeability, Systemic Inflammation, and Macrophage Dysfunction. So that pretty much summarizes what the authors the findings of the paper. Dysbiosis, I guess this is a term that I've heard for the first time in this paper. I guess it means a dysregulation of the biotic environment of the intestine. So a microbial dysbiosis. And here is I'm not sure if I can get this much bigger, small graphical diagram. Let's see if I can click on this. I guess I can't. Can you guys see that? A little graphical abstract? Is that kind of clear?
Yep.
Yeah. Anyway, so it's just basically a little graph showing what is happening in young versus old. So we have this little red blood vessel. We have here macrophages in purple, and we have the epithelial lining of the gut here, and then the lumen and a whole bunch of little microbes floating around. And in dysbiosis, we could see that there's greater or enhanced permeability of the gut microbiome that is letting things through. And as a result, you have increased levels of inflammatory cytokines such as TNF or tumor necrosis factor and IL-6. And this overstimulation leads to, ironically, a decreased killing by macrophages over a long term. And also,
Oliver, one thing you missed, it's not that the dysbiosis of the gut microbiome and then the next step is an increase in inflammatory cytokines circulating. It's that there's an increase in gut permeability, and then there's an increase in circulating microbial products, which then probably activate inflammation. So
yes,
that's a very important point.
Yeah. Okay. So the permeability increase. Right. So you have increased microbial products like lipopolysaccharide, et cetera, which promote further stimulation. Is that correct?
Yeah. They bind to things like TLRs, and that stimulates inflammation. Yeah.
Yep. Okay. So on to the results. So TNF drives age-associated defects in macrophage function. So the way this paper is structured, I have to scroll all the way down to get to figures. And this is figure 1. So figure 1 through J. So basically what they're showing here, and we'll go through some of these subfigures, is that inflammatory responses increase with age. I think we've seen that in a number of other papers, a number of other results that have been presented. And what you see here is a number of different assays, figure 1A and 1B. It's a bacterial viability assay. So this is looking at the killing power of macrophages that have been isolated from either young or old mice from the peritoneum or the bone marrow, right? So, killing of S. pneumoniae. And you can see that the killing power is decreased in old mice. So you're having some negative consequences there. And figure C, this one I was a little, I don't know, no control there. I'm going to have to take their word for it. It's a tetramethylrhodamine labeled S. pneumoniae, and they observed macrophages derived from old mice but not young mice, ingesting it but not degrading it. So I'm assuming this is supposed to be a little S. pneumoniae that's engulfed but not destroyed. I'm not sure where it is. It's just one set of images that is claimed to be representative. But the rest of the figures look at various cytokines that are in the blood. And here we see tumor necrosis factor, young, being low, about roughly 1 picogram per mil. And then this is enhanced to a little over 3 picograms per mil in old. These are correlated with inflammation, along with IL-6, interleukin-6, picograms per mil. We have young at about 3 and old about, I guess, 7. And these are two different, so E and F are looking at, one is in whole lung homogenates and one is in blood plasma. So you do have an increase in inflammatory molecules floating around in various areas. You have decreased killing power. Here is just a histology section of, I believe, lung tissue, and looking at these stained, which are believed to be pink macrophages that are infiltrating. So this is H&E stain of formalin-fixed sections of lungs. And this is just the quantification of what we're seeing in G. So HIJ, I'm just going to summarize that. So these are lung slices. So they stimulate with either heat-killed S. pneumoniae or lung culture, and I believe macrophages that are, no, just lung slices. So they're stimulating either with PBS or S. pneumoniae. So you see IL-6 levels the basal levels are pretty high in old, but they do go up. What's I? I is whole blood, and J is IL-6 production specifically for macrophages derived from young and old mice following 24-hour stimulation of either PBS, LPS, or S. pneumoniae. So they are responsive, but the basal levels of these inflammatory cytokines are up. So the take-home message of Figure 1, if anybody wants to interject and say anything or add anything to that, is that the inflammatory responses go up with age when it comes to the measurement of TNF and IL-6 in old mice, and that the killing power of macrophages is decreased. Is there anything else anybody wants to mention about this?
Nothing really new there, right? They're just showing what's already been known. What's interesting is that why would they focus on respiratory, the lung? Why would they focus on the lung and lung macrophages? And they could have shown any tissue, muscle, brain, liver, right? So nothing special. It's just the lab that publishes paper. That's their specialty, so that's what they focused on. But I wonder what the data would look like. How would muscle macrophages look? How would the killing power there look, or in the brain, or in the liver? So that's for other labs to pursue, I guess, right? So.
Yeah. Okay. So that sets a kind of a foundation. There's certainly a bit of replication there, but it's good to see that they're able to replicate the results that everybody has grown to expect. Okay. So back to this paper. Let's go to Figure 2. Screen share. All right, we're back. Scroll. Figure 2. Chronic exposure to TNF contributes to increased inflammatory responses and tissue damage that occur with age. So now they are directly correlating. I'm reading basically off of the figure legend here, correlating levels of TNF or TNF activity to increased inflammatory responses and tissue damage that are occurring with age. And I just wanted to make a comment here about this figure. They have, so this is another one of these killing assays with viable bacteria, S. pneumoniae, and isolated macrophages from bone marrow. And this is done in a petri dish. We have young mice. You have increased killing power. Old mice, decreased, but still some. If you add TNF alpha, it seems to inhibit it, and old with TNF alpha is even more inhibited, the killing power. This TNF alpha is something they add to the dish, to these macrophages, and I looked at the concentrations. I don't do these assays, so I don't know what are the appropriate amounts. They're adding 10 nanograms per mil, so that is 10,000 times higher, if I'm not mistaken, than what's found in blood plasma, because we're dealing with 1 picogram being the normal per mil of TNF or 1 to 3 picograms if you want to include the old cohort in the blood plasma of TNF alpha, I think. And in here, they're adding essentially 10,000 picograms per mil. So I don't know can we make a comparison here? Is this a fair comparison? Anybody want to chime in on that?
That's a great point, right? Is super physiological levels, what would it look like at physiological levels? Maybe it would have no effect, right? I think it's a very important point.
I mean, that's like orders of magnitude more.
Yeah, for sure.
Exactly 4 orders of magnitude.
Yeah.
So anyway, be that as it may, that's the result when you add in 10,000 picograms per ml. So next in B, I'm like old wild type mice, so now they have these mice that are TNF alpha knockout mice, and they show that they don't have elevated levels of plasma IL-6. So here we have young wild type, old wild type. Levels are high, and so we have about young is about 2-ish, and then old is about, I guess, 7-ish picograms per mil, and they are both in old and young repressed to about, I don't know, less than 2, maybe sub 2 picograms per mil, and the old is certainly, it's much more dramatic. It's interesting, again, my immunology knowledge is a bit fuzzy going back many years, but from what I've done reading-wise, IL-6 inhibits TNF alpha. Is that correct? I believe it does. So, I mean, that would make sense. If you have less TNF alpha, then perhaps the levels of IL-6 go down accordingly as a response. So, just pointing that out there. C, so figure C. They're looking at IL-6 production in the whole blood of young and old tumor necrosis factor knockout mice following stimulation with either LPS or just PBS. So young TNF knockout, old TNF knockout, no stimulation, with stimulation, no stimulation, with stimulation, and you do have a response, you have an increase in IL-6. Demonstrated that old TNF KO mice do not have higher inflammation compared to young mice. So there is I guess, some sort of correlation with TNF levels, perhaps. Like I said, we do know that IL-6 is an inhibitor of TNF. So other than that, if anybody else has another correlation to make, I'd love to hear it. And in D, they're doing basically whole lung, again, lung tissue homogenates, showing that the same result, there's no higher. In lung tissue, what do we have here? We have old TNF knockout. Yeah, no higher. But this is not in response to any stimulation with the LPS. So these are just basal levels. So again, seems that the levels of IL-6 are required. Higher levels require presence of TNF alpha. And E and F basically are showing that there's no differences in killing power between macrophages and old and young TNF macrophages in cells have been, mice have been knocked out for TNF alpha. So, anything else we can take home from this other than that chronic exposure to TNF contributes to increased inflammatory responses?
So I have a general comment, just not in terms of figure 2, but so if you notice, they open the title of the paper as age-associated microbial dysbiosis, blah blah blah, but yet they start with the inflammation story. You know, they don't even follow their own, you know, their own title. They should have shown figure 1, microbial dysbiosis, which they show later, and then figure 2, intestinal permeability, figure 3, inflammation, figure 4. You know, it's just interesting how they frame this.
Yeah, it is flipped.
Yeah, they could have told the story better. Not that it's told badly, but you know.
Yeah. Let's go back to this paper and see how the story develops. All right, let's go. So. Figure 3. I think we're getting more interesting now. Okay. Okay, here we go where it starts to get more interesting, which is figure 3, the microbiota increase, intestinal permeability, age-associated inflammation, macrophage function, and longevity. Michael, I think that's a little better, right?
Yeah.
Okay. So they're doing permeability studies using a number of these different assays. And basically when they talk about permeability, they're talking about things getting passed from the lumen of the intestine, which is, here's a cross-section of the lumen, which is this white part, to pass these microvilli into the bloodstream and back here. So there's a number of ways things can get through. One of the ways is sort of passively, that's up through a paracellular process, which is basically in between cells, right? You have, you know, the cells are connected together with a number of molecules that basically attach themselves, like cadherins, for example. And these interactions, if they weaken somehow, you can have permeability occur. And the way they measure permeability is they either have this FITC dextran molecule, which is, you know, they use something that's indigestible and something that is can then also therefore pass through and be measured in the bloodstream or in the serum. And dextran is this large molecule that's conjugated to a fluorophore, so they can measure fluorescence. And then later they use this chromium 51 conjugated with EVTA, which is also passes through somehow. I pulled up an old paper. Do I have to click on this, or do you see intestinal permeability of chromium, EDTA, and infectious diarrhea? No, I got to click on that. Okay, so do I have to stop share and then reshare? Let's try this here. Okay, share screen. Yeah, good. So I just wanted to see how this chromium EDTA assay works. This paper is from '93, and to be quite frank, they're not sure exactly how it gets through, but somehow it manages to pass through the colon and enter the bloodstream. But it's not really clear how this permeation is exactly occurring. But anyway, stop sharing that. Go back to screen share and go back to paper. Okay, so those are sort of, I guess, the accepted techniques that are used. And you can see in figure 3A that permeability does, in general, increase. And there's a wider and wider spread with ages to this FITC dextran fluorescence that's found in, let's see, is that blood serum? Yeah, translocation to the circulation following oral gavage. So they basically fed the mice this. And in B, when they look at young and old mice, they don't seem to have any detectable changes in either epithelial architecture or inflammatory infiltrate. But I'm not sure if histologically you can see that. They do look a bit different, but I'm not a histologist, so I don't know if that's abnormal or normal. Then they do mucosal to serosal flux, which is basically from, I guess, the lumen to the bloodstream. Chromium. The only big difference between fitseed dextran and chromium is that they're also two different sizes. One is much tinier, the chromium, than the fitseed dextran. So they're looking at, I guess, a size range of things being permeable. The ileum is basically part of your lower intestine, the colon. Your upper intestine, your small intestine, and your large intestine being the colon. They don't see much translocation through the ileum, but they do see it go up in the colon with old mice. So the permeability definitely increases. They also look at blood plasma levels D of a bacterial peptide. So this muranil dipeptide nanograms per mil, and you can see that perhaps as a result of this permeability, you have these peptide levels, circulating peptide levels go up in old mice. And any comments on this thus far, halfway through this figure? Okay, so far we can see that inflammatory responses with regards to IL-6 and TNF go up. We also see that permeability increases in the intestines of mice. And now they start looking at germ-free mice that have been raised in an environment, as sterile or aseptic as possible. I've never bred these mice, so I don't know exactly the protocol for doing so.
Hang on, Oliver. Maybe I just have a minor point. So yeah. The MDP, the muramyl dipeptide, so lipopolysaccharide is what people normally look at when they're looking at gut microbial markers in the blood. And just as a point of reference, it's a marker of gram-negative bacteria, so things like E. coli are gram-negative. The interesting thing about muramyl dipeptide is that it's found on both gram-negative and gram-positive, so things like Staphylococcus aureus, which are gram-positive, and obviously are different from the gram-negative E. coli. So MDP, muramyl dipeptide, you're basically covering both of the major forms of bacteria that are found in the intestine, the gram-positive and the gram-negative, which would suggest that considering LPS has been found in the blood of older adults versus young, now you've got MDP, which is up in old versus young. You could make the argument that it's not an effect, the gut permeability effect during aging and where it increases, it's not necessarily an effect of gram-negative translocation through the gut. It potentially is gram-negative and or gram-positive. So it's almost like a non-specific permeability issue.
Yeah, except we don't, since it's working on both, we don't know if it's the effect on gram-positive per se, right? Is there a gram-positive specific marker that could be measured?
Yeah, I mean, the lipotechoic acid, which is found in Staphylococcus aureus, which is a gram-positive.
So why would they go from LPS to this one if it wouldn't clarify sort of what you just suggested?
Yeah, I'm pretty sure that their group has measured MDP in other papers. They probably did it for convenience. But ideally, in not just bacteria, there's fungi and viruses, bacteriophages, all located in the gut. This is an understudied and underexplored area. What exactly is translocating? Is it only bacterial products? Is it fungal products? Is it viral products? What's going through? So when I see that, that's what I think. I think these are things that can be looked at in future studies.
Yeah.
The groups. So.
All right. So they're laying the groundwork. Okay. Let's share the screen. Let's go back to the paper. All right. So now they're looking at permeability and old and young, but in this case, now they're switching to mice that have been reared germ-free. So now we're seeing some more interesting comparisons. Now, to me, figure, what is this, figure 3? Figure 3E is sort of important, sort of, I don't know, one of the crux kind of figures to me. So mucosal to serosal flux of chromium EDTA, it goes up, and they say this is significant. I believe wild type slightly here from like 0.2. I don't know what the measure is. I have to take a look at the again. But whatever, it's from 0.2 to 0.3. They say this is not significant from young to old, from 0.2 to, I guess, 0.2-ish. I don't know. I mean, maybe I'm being nitpicky, but when I see things that close, I'd like to see more numbers thrown in there, because E says N equals 5 to 8. I don't know if that's 6 or 7 or 5 and 8 or exactly how many, but that's how many, or young and old mice, or if that's 5 to 8 combined. So it's a small number of mice, and I don't know. Just me personally, I would have liked to see more mice to see if this really is not significant. I don't know if anybody else has a comment about this, because this seems sort of crucial to the rest of the results, right? I mean, anyway.
Yeah. So I don't necessarily have a comment on that. I mean, that's they didn't put the p-value there, did they? I mean, what was it 0.06? Was it 0.1? How close was it? But then also, their definition of old, where they use 600-day-old germ-free mice, average lifespan for C57 is 900 days. So this is, and maximal is 1200. So this is basically half lifespan. This is the equivalent of a 60-year-old. I mean, is 60-year-old really that old? I mean, the 28-month-old mice, if they get closer to 800, 900 days, how would these effects look? Maybe even worse. So in the interest of cost, of course, 600 days is cheaper than 900 days. But when I see these data in old as 18 months or 20 months, it's somewhat laughable. I've worked with 30-month-old mice, and those guys are old. I mean, so.
Yeah. So, for me, it's just, the error bars are kind of getting a little close, and, if I just like to see, more mice dumped into this to see if there's going to be a change or an alteration. But anyway, we'll take the p-value at face value and say that there is no significant difference, and that germ-free mice, according to this, don't have a significantly different permeability. So there is a correlation between being germ-free and being not germ-free, assuming the same genetic background, that there is a correlation between permeability of the gut and having a germ-free microbiome. And perhaps as a consequence, you see a survival curve here only going to 600 days, but you start to see mice die off, and this is a population of, well, I don't know what the population is here. Whatever the population is, it's significant. I believe that's what the little asterisk is, and versus old germ-free, right? So old in quotations. And then they start looking at other aspects of inflammation, which is the IL-6 levels. And this is young wild type versus old wild type, and then young germ-free versus old germ-free. And there doesn't seem to be an increase in IL-6 levels for the old germ-free. I just want to scroll back, bear with me, to IL-6 levels here. Well, I guess that's about the same. Old, 10. Yeah, okay. So yeah, there doesn't seem to be, so IL-6 levels seem to be fairly constant up to, I believe that's 600 days. And then histological sections doesn't appear to be increased leukocyte infiltration with age for old germ-free. So these are just the numbers from the infiltration of total lung area. So this is just the number, quantification of this qualitative data here. And I is looking at lung homogenates, and J is looking at IL-6 production. Well, actually, let's hold off on this. IL-6 levels in lung, again, seem to be not significantly going up with germ-free. And in J, we have stimulation with PBS or lipopolysaccharide, and this is IL-6 production in whole blood of young and old, wild type. SPF is this, I guess this was a standardized, I'm blanking on the acronym here. Some
specific pathogen free.
Yeah, specific pathogen free. I guess they were reared with a probiotic blend. Is that correct? Or some sort of biotic?
Yeah, they're not germ free. They have some microbes, but they're defined. I just don't know which microbial species they contain.
Yeah, it's like air mice, Oliver, at MMTP. They're in a SPF facility, which is fairly germ free. As I mentioned to you before, that just to go back to the lifespan curve, it's consistent with another study where they found that SPF mice do tend to live a little bit longer.
There's actually a couple studies that have looked at germ-free mice versus wild type, and the germ-frees live longer.
Yeah.
Full lifespan curves, not this 600-day stuff.
So, I mean, at best guess, I would say it's certainly going to be rounding the curve anyway. So, health span, if not massive increases of lifespan, germ-free, the less sort of germ burden you've got, microbial burden, the better off it is for lifespan. Kind of makes sense, really, doesn't it?
In those conditions. I wouldn't say that in the real world.
Yeah, I mean, it's going to be very difficult to live in a bubble, really, isn't it?
Yeah, literally, yeah.
Yeah, I mean, you couldn't take out pizza or anything, for instance, Oliver. Yeah. You couldn't call out for Domino's, which is unfortunate.
Okay, so let's take a look at J here. Stimulation with LPS. So PBS, basal levels. We have young wild type, IL-6 picograms per mil. Stimulation. Let's see, what is this? Is this 50 picograms per mil? We have different amount of picograms per mil up there. IL-6 production in whole blood. Plasma cytokines, germ-free. I don't know. It seems like there's like almost 1 order of magnitude difference here, right? Am I reading that right between G and J? Like, why do these mice have 50 picograms per mil and these have 2? It seems to be a big difference, doesn't it?
Oh, J is they're stimulating with LPS.
Yeah, but PBS is not. That's just their basal.
Oh, right.
Because here in G, when they look at wild type versus germ-free with no stimulation, young, old, the old jumps to 7, but here the young is already baseline at 50. I don't know if that I don't know if mice have that much of a fluctuation or difference between IL-6 levels, or maybe different cohorts at different times have that much of a range of an order of magnitude per baseline. I'm not sure. I haven't looked.
I don't know.
But anyway, these are numbers that be good to have an immunologist maybe say, oh yeah, that's normal, or yeah, I've never seen such a fluctuation before. I don't know.
Yeah.
Anyway, so baseline is much higher with old wild type. Young germ-free and old germ-free seems to be similar baseline. Again, they say it's not significant from wild type. LPS stimulation, again, you have a greater stimulation with old wild type as well as young germ-free and old germ-free. But the young and old germ-free stimulation, and also the old germ-free, is similar to the response you get from LPS stimulation for young wild type, which I guess perhaps is not too much of a surprise since the baseline levels are low in the old germ-free mice to begin with prior to stimulation. And then K, we're looking at macrophages from young and old germ-free mice. So unlike what we saw before, there's no difference in killability, I guess. um, counting viable S. pneumoniae bacteria. And L is, again, after, so IL-6 production, so after stimulation, so these are macrophages from, bone marrow macrophages. The IL-6 production is very similar stimulation-wise between young and old. So I guess the take-home message of this entire figure is that looking at just those parameters, young or old germ-free mice behave like young wild-type mice that have that blend of microbes growing in their gut, the pathogen-free microbes, specific pathogen-free. Any comments, anybody on that?
So the J really says something. I mean, so in the presence of LPS, obviously old wild type, whole blood, there's an increased production wherever it's coming, whichever cell types it's coming from.
Right.
So if you consider that old blood has more circulating microbial products, just using their MVP example that they showed earlier, and that the germ-free doesn't, I wonder if LPS synergizes with other circulating microbial products in old wild type, so that the IL-6 producing cells in aged animals basically freak out. They're like, this is ridiculous, right? We're going crazy here. And they overproduce an inflammation, or if it's just a specific effect of LPS alone, and then these cell types that produce IL-6 basically freak out, overshoot their inflammatory, what they need in terms of inflammation. So, because the IL-6 producing ability should be the same. It's, they're both getting the same dose of IL-6, of LPS, whether they're old germ-free or old wild type. It's still the same dose of LPS. It's just the old IL-6 producing cells just freak out, right? Why? What's causing that?
Yeah. I mean,
is it because of other stuff circulating? Is what would it? Yeah.
I mean, it is a proportional response. So if we look at young wild type and old, it goes from about 50 to, let's say, 120. And if we go from, well, actually, that's the background. If we go from young to this, it goes, from 50 to about 120. Or it go from 120 to about 240 or so. So it is about a 2x, twofold response.
Do you figure that the germ-free old should have the same IL-6 producing ability, yet it doesn't overshoot it like the old wild type?
Yeah.
Just why that is, I have no idea. Whatever mechanism that may be, but it have a funny visual. It's like, the IL-6 producing cells in the blood. It's basically the lights are off, and then an intruder comes with the lights off, and then they're just flashing their flares trying to figure out where it is, and the whole room gets lit up. The analogy being inflammation is the light up, but it's just interesting how the old germ-free don't do that.
Well, perhaps they're primed as a result. It'd be interesting to see if just having maybe let's say you have germ-free mice and you constantly stimulate them with just LPS, will you get the same IL-6 response? Right.
But don't forget, macrophages also produce IL-6. Depending on their polarity, you've got M1 and M2, which are the broad two types. M1, which is the aggressive, inflammatory, attacking sort of macrophage, that produces lots of IL-6, and the M2 produces other factors that are more sort of calming and mediating for tissue repair. In fact, that's why they've had success switching M1 to M2 to actually facilitate repair. So, inflammation breeds inflammation. So, it's going to be a sort of avalanche effect.
That gets me into maybe the balance between the M2s and the M1 is different in the germ-frees versus old wild type.
It could be.
Right, and you predict a higher amount of M1 to M2s in the old wild types versus old germ-free, which may have it reverse more M2s, but I mean, that's, you know, yeah.
But yeah, certainly M1 macrophages generate lots of IL-6, so it just keeps going round and round and round and round in a circle. So you know, where does it stop?
Yeah,
that's the problem, isn't it? I think inflammaging itself just becomes a self-perpetuating feedback, which is bad news, really, for us.
All right. Well, with that, we are getting to the end, actually. I'm going to need help with Figure 5, their analysis of the data, and also the microbiome here. So Figure 4 is looking at the actual microbiomes of young and old ASF mice, and also, I believe, do they have germ-free mice here as well? No, actually not, if they're germ-free. So they're looking at mice with minimal ASF-derived microbiota were aged, and their intestinal permeability measured, so it goes up.
Hang on, just to jump in real quick, Oliver. I think it's an important point to say why they used ASF mice. So they hypothesized that just having any microbiome would lead to an increase in intestinal permeability and increase.
Right.
So they figured these ASF mice have very low microbial diversity compared to the germ-frees and compared to wild type. So they figured just having any, let's see what happens.
So just by having any, and in this case, the ASF, they see an increased permeability, and they see increased plasma IL-6 levels, and they see also prior to PBS stimulation, also increased IL-6 levels, but also much greater response to LPS, lipopolysaccharide stimulation. And they look at, so figure ABCD is the taxa summary of microbiota of young and old ASF mice. So you see that the proportions change, and these are different, I guess, phyla and genuses and orders. So you have young and old, and this is just showing you that basically the percentage spread of the different types or different classifications between young and old. Let's see right here. E. Taxial summaries illustrate the composition of the young and old microbiota that's retained upon transfer to young or old mice or for independent colonization. So here they, so these are, let me see. How does this compare? If you help me out with D and E, why is this different?
What D is, you can just see the compositional changes young versus old in the ASF mice. I mean, you can see the pink, right? And then the pink is, you know, whether Bacteroides is the phyla. It's just dramatic. They're just showing that the composition changes with age. Right.
But is the difference between young and old and D different from the E, young and old?
What are they doing here?
I mean, they have different designators here, so I'm not too familiar with this nomenclature. I believe it has something to do with like phyla, genus, order, family, but these are in uppercase with colons, and these are lowercase with an underscore, and I'm not really up on my
so the G, whether it's uppercase or lowercase, those are genus.
Yeah, but why is
I don't know why they did that. Family Lacnospira, however you say it, that's the family, whether it's capitalized or lowercase. I just know in E they did fecal transplants, but yeah, I don't get why they've got young and old SPF and then recipient donor. I think actually the left side of E is actually in the right side of E. They just put them next to each other.
So these recipient and donors, is this fecal transplants or this just in the box together?
No, they gave them fecal transplants. Young to young, young to old.
Right. So this, because they mentioned somewhere that, that's G, right? They just colonized the mice together. This one here, I guess, because here they say colonization of, oh, okay, never mind. They did do fecal transplants. All right. Just got a little lost there. All right. So here we have different recipients and donors. So basically young to young, old to young, vice versa. Young to young, obviously, we have the same thing. Old to young, you have a composition that if you have the recipient is old, young to old, if you have the recipient that's old and you transfer the young composition, which is shown here on E, and you do the sequence analysis. How long did they keep that there, though? They do the transfer. Can't remember.
I don't remember either. It's usually about a month, but it may have been shorter here. I know it should be there somewhere.
So when they transfer the young, it remains young. Interestingly, when the recipient is young and they transfer the old, we have colonization with old.
It says 6 weeks. After a minimum of 6 weeks, well, they measured the changes in paracellular permeability, but since they put that data together, I'm guessing the compositional analysis is similar to the paracellular, what they measured for paracellular permeability.
Right. So in this E, basically what we're seeing is during the length of the experiment here, the composition of the microbiome was reversed essentially for both young and old, vice versa, when they did the fecal transplant.
But what about compensatory mechanisms here, I'm thinking?
Well, I mean, they're just showing this. There's nothing, I mean, that's the only data that so far we see. I mean, I don't know what mechanisms there are. They just did a fecal transplant. They're showing what you see after a month. F is, now they're looking at permeability measured in germ-free mice colonized with young and old microbiota. And with old microbiota, it appears to be that the permeability is, I guess, shifted up using a chromium assay. And in G, it's, again, a chromium assay, and they are a colonization of young germ-free mice with old. And we are looking at permeability, so young, it's low. Donor old to young, we have, again, increased permeability. Young to old, I don't know what they say there, but it appears to maybe go down a bit, right? He is, however, old mice colonized with either young or old demonstrated increased permeability. I don't know. I mean,
yeah, small trend. I mean, but basically it's going to be very hard to rescue old microbiome, even with young stool.
Yeah, I don't know if I would firmly say that, okay, it's having no effect.
Yeah.
You know, but anyway. So in H, they're looking at circulating TNF from all the young germ-free mice colonized with young microbiota, or young recipient, old recipient colonized with an old GF mice colonized with young microbiota. So what are they saying here? You have a young recipient with old microbiota, and an old recipient with young microbiota. Is that correct? Am I reading that right? So, and this is circulating TNF levels, picograms per mil. All the young germ-free mice and old GF mice.
But the question is also why do the mice have a circulating TNF that is higher? I mean, you need to separate what is the cause from the microbiome and the other factors also. It's not very clear.
Definitely.
Yeah, so this is I don't know, this is kind of weird. This is so this is N equals 13, and this includes 5 mice colonized with young microbiota, 5 young mice, and 8 colonized with old microbiota. Why would you lump that together? I don't know. I'll go back to this, but I seems to separate this out. So the recipient, young, This is TNF picograms per mil. We have young and the donor is old, so you have greater circulating TNF levels. Young into old seem to have maybe, I guess, about the same high levels, and old higher. And there's a lot of overlap with these error bars here. Seems to me old. Well, what are they saying about the significance of this? Again, these are. Once I see overlaps with error bars like this, I'd like to see more animals thrown in there to get that out of the, move that out to see if that's gonna still, that effect is still gonna be there, because there does seem to be an effect with when the donor is young going into old, because TNF alpha levels are, even though this might be not considered statistically significant, it seems the trend is lower, and we see certainly a trend of permeability, even though it's not statistically significant, it seems to be lower. So taken together, there might be some beneficial effect from a young microbiome. I don't know, but that's not what statistically is claimed here. Okay.
That kind of ties in with the transfusion, like the blood transfusion data, doesn't it? Where old blood into young and then young blood into old, there's just a minor dampening of outcome-related effects, whether it's muscle, injury recovery or whatever. So it's a similar effect going on with the microbiome. I mean, I totally agree they needed to increase that N, old and young, recipient and donor, to flesh that out. But I'd say it's consistent with the blood transfusion data, young to old or old into young, or the parabiosis stuff.
Question is there also what has just to do with like dilution factors? And
yeah, could be that too.
Really, you want to see at least N equals 40 to reduce statistical noise. So I'm not quite sure how many they used here, but I thought I'd call 8 mice. Statistically, that's going to open you up to quite a lot of noise. So you really want N equals 40 at the minimum. 50 or more is better. But from what I can ascertain, it does seem to suggest that if you take young microbiota and put it into old animal, certainly here it seems to have at least some reduction of TNF alpha, so which is good. And that's consistent with some other studies we've seen. But as you say, I think it warrants a larger amount of mice, really.
I don't know about 40, but for sure when you see an effect, the first thing you're going to say is it real? You've got to repeat it internally in your own lab. So you've got to increase the N, that they would just get away, I mean, to be critical with eight mice and that, that old recipients and young donors, right? That it wasn't repeated internally. Hey, add another 8. Let's see if you get that again, because you don't want to publish this and then it's like, well, it's not a real effect, right? You want to make sure your effect is real. So I don't know about 40 from the start, but definitely that should have been a lot more. Yeah,
I mean 8, and with sort of 8 or 10, you're going to get outliers that dramatically affect affect it. So the bottom line really is, the more mice you've got, the more sort of gentle any outliers are going to have an effect on.
Yeah.
So we reached about N equals 40 for MMTP, and it was reckoned to be a very good sort of number to eliminate statistical noise. 40, and we consulted people like Richard Miller and Spindler to reach that sort of total.
Yeah.
So we've got 40. We've got a control group of 40, and we've got a test group of 40. So we're not messing about. So essentially, what we wanted to find out was if there was a trend, we can sort of say, hey, look, Senolytics is definitely doing this.
Yeah.
So I think 8's a bit small, maybe 20, but as you say,
15. I mean, double the N, or triple. Do that same experiment 3 different times. So at least get to 24. So you believe it internally, right? Always more mice is better. 40 is good, 40 and 40, but then it becomes a price cut, issue. And they've already got a ton of stuff that costs them money here, aging the mice to 600 days.
Yeah, that's true. I mean, we encountered that because we bought mice. We had to obtain mice that were a minimum of 18 months old, and that gets expensive. It really
Yeah.
does, especially as we're doing a full lifespan study. So we're not obviously, only doing it for 600 days and then calling it quits. It'll run until the very end. So it's
Yeah.
expensive,
Yeah, definitely.
All right, so last figure here, and the data is a little bit more, I guess, cleaner. This is in mice that are old and young, germ-free, and also our TNF alpha knockout mice, young and old. And they're looking again, leptoplasma, FITC, dextran, permeability, gut permeability, and also circulating levels of the muromyl dipeptide, which, as Michael's pointed out, is found in both gram-positive and gram-negative bacteria. And basically what you can see here at a glance is that knocking out TNF basically drops the levels for both the old mice to young, young germ-free or young wild type, implicating TNF in perhaps permeability. I don't know. Did they? I would say that did they do the I guess the inverse of this experiment, which is inject mice with TNF and see if the permeability goes up?
Didn't they? I don't know. Figure 1. Did they do any of that? No.
They added 10,000 times more TNF into plates. That was the killing power here of. Was this the one, figure 2? I think so. Yeah, 10 nanograms per mil exogenous TNF.
Well, in their defense, chances are when TNF is up, you're going to get IL-6 up, CRP up, not just one pro-inflammatory cytokine. So sure, they used, orders of magnitude super physiological levels of TNF, but when you combine TNF with all the other age-related cytokines that are up, is it then physiologically relevant at the dose? You know what I mean? They use the bolus. Does that mimic an age-related bolus of inflammation? I don't know.
The other issue is I should have read this a little bit more carefully regarding the methods. But these knockout mice are they conditional or are they because you're going to get completely different results if they're knockout mice bred from the beginning or they're conditional knockouts later in adulthood. I mean that was a take home from experiments done years ago that you'll get opposite effects.
Yeah.
So I'm not sure if these are knockout mice bred from the get-go or if they're conditional knockouts, meaning that they're knocked out later in adulthood.
Yeah.
So that's obviously going to, you might get completely conflicting results depending on which one you use. Well, I'm not going to go into this statistical analysis here. If somebody wants to tackle the principal coordinate analysis based on Bray-Curtis and showing how this all diverges, I kind of will take this data at its face value here. But basically, what we're, the take-home message from all of this is this little graph that's on the bottom, is that you have intestinal permeability, and that causes bacterial products to translocate, and this is not happening in germ-free mice. You have systemic inflammation, which is inhibited by TNF knockout, perhaps, and then that causes an increased permeability, and you basically have this kind of positive feedback loop that happens.
But something I'm thinking about, why don't they measure all the different types in the TNF family? I mean, they should look at the different.
I don't know. The other thing about the gut microbiota is obviously there's a lot of stuff going on. And this is just one paper, if I got the right one here, that came out recently. Is that the one? No, that's not the one. Stop share. Sorry. Share screen. Yeah, this should be it. Where's that paper? Are we seeing a urolithin paper? Oh, there it is. Nature Medicine, urolithin A induces mitophagy and prolongs lifespan, and C. elegans increases muscle function in rodents. So what does that have to do with the microbiota? Well, urolithin A is a compound that is basically derived from another compound in pomegranates, and it induces mitophagy, has great responses, but only in people that have a certain microbiome. Only in rodents have a certain microbiome that can convert the precursor to urolithin A that's found in pomegranates to the active urolithin A. So this is also tying in, obviously, into longevity, and obviously through diet, but also to the microbiome, because you have to have a particular microbiome to efficiently have the microbes essentially convert this compound, not your liver, but the microbes in your gut to the active urolithin A. I'm just tossing that out there just because there's a lot of stuff happening with your microbiome, not just inflammation, but perhaps also how certain dietary compounds are processed, which can then feed back into inflammation because a lot of these compounds are anti-inflammatory. So
polyphenols, for example. Yes, I'm going to talk about polyphenols again. But yeah, I mean, I've just been looking at a paper or about 60 papers that talk at great length about quercetin, which is my favorite polyphenol. But it very much influences gut microbiome. So yeah, I think you're spot on, Oliver. I think diet is a big influencer on what goes on, and certainly there's good evidence to suggest that if you eat a certain type of diet, you'll get a certain type of bacteria, and vice versa. If you change your diet, that changes.
Yeah, I think we need a lot more experiments that can critically tease apart all of these influences, because there's a lot of things that are overlapped, and when you have too many variables like that overlap that you don't take into consideration, you'll end up with big error bars, and it starts to get a little bit murky. Well,
I can tell you where I see this whole thing going with the microbiome stuff, and so the microbiome is the composition of the microbiome isn't just going to be affected by diet; it's going to be affected by host genotype.
Mm-hmm.
So, for example, OB, the leptin deficient mice, actually have a different microbiome compared to the wild types. And when you transplant the gut microbiome from OB mouse, it transfers the obese phenotype. So host genotype, you're going to have a certain microbiome. So we've got to take full DNA sequencing, human genome, and then you can predict what your gut microbiome could be to optimize your host genome, to minimize disease risk and maximize longevity. And then you can add diet in there to further, add to those effects, how to truly optimize it, whether it's through diet, whether it's through exercise, whether it's through supplements. So, but we're going to need, Alex Zavar, however you say his last name, Zavaronkov, and his AI crew to get our host DNA, to get our microbiome sequence, to get how much exercise, how much diet, which specific types of diet based on your genotype, and crunch all the data to get us on the low risk track for aging and disease.
So are we entering the era of everything omics?
I hate to sound like the typical scientist. This is over 10 years away, but I'd say within
everything omics.
It's coming. It's coming faster, rather than later. Aaron Eliav's group, who published some stuff on the glucose handling with the white bread meal, and depending on your gut microbiome, you can handle a white bread, which is just a simple sugar and insulin response. If you have a certain microbiome, you can handle it better than not. So I guess that's kind of on the road, but yeah, within 10 years, I'd expect this AI to crunch everything, all of the omics, to take off. Those in the know will be able to use it, whereas the masses may not be able to use that stuff for a few years. But if that whole approach isn't done in the next 50 years, I'm going to be really mad, because when you really think about it, to truly minimize disease risk and maximize longevity, you've got to have all this big data put together. Otherwise, we're just guessing, right? I
think we need a better instant messaging app. That's what I think we should focus on.
That's
No, I don't think so. But I can definitely see the direction things are going in the way of sort of almost like customized probiotics. Like you get your probiotics drinks and all that. I think you'll end up having a genetically customized probiotic to suit you.
So before that would happen, though, unfortunately, most of the microbiome field is focused on bacteria in the gut. But the gut composition, it's an ecosystem comprised of bacteria, working together with fungi, with viruses, with bacteriophages. So just the full gut microbiome composition and how they're working together, I mean, it's not even being done. I don't think I've ever seen this. Well, there's a handful of studies that look at fecal bacteria and how they relate to fungi or fungi and viruses, but all of them together, this is your ecosystem and how it looks. So and the reason that's important is because if you take a probiotic and it affects your bacteria, well, how does it change your overall microbiome for the positive or negative? So, but then that comes back to, you need some AI modeling to model this stuff. It's just so much data, trillions of data, so yeah.
I'm waiting for whole genome editing of all my trillions of cells to mimic that of a supercentenarian so I can smoke and eat and drink whatever the hell I want.
But supercentenarians have atrociously bad health. They have better health compared to 80-year-olds, but compared to a 20-year-old, I don't know that I would want, supercentenarian with, albumin levels of 3 and red blood cells of 4, and they're basically suffocating to death because they don't have enough red blood cells to carry oxygen. So I don't know if I'd want to replicate that necessarily.
Although there was that clinical study last month in China where they did a very large study of super healthy people that are in their like 90s, 100s, and they compared the microbiota with people who were ridiculously healthy in their 30s, and they were very, very similar.
Yeah.
So that says something about the gut. And as I say, it does tie in with that 2015 Newcastle study as well, where they showed that one of the criteria for living 100 plus was low inflammation.
Yeah.
So it kind of ties in.
I would be a bit careful there about China because they lacked the birth registration 100 years ago. So these ages could be inflated.
Or deflated, right? They're not 100, but they're 80.
But I mean, that's a fair point. But you can tell whether someone's older or, younger to a reasonable level. So
to put those two studies together, the inflammation in microbiome. So the microbiome in the centenarian is not being significantly different versus the young, right? So based on the paper we just reviewed, right, that Oliver covered so well, if the microbiome is like a young person, your gut permeability is solid. You have less leak into the blood, and if you have less leak of microbial products into the blood, less inflammation. So it all ties back.
Yeah, and of course, if you're going to have less inflammation, it also ties into the whole macrophage situation. So you're going to have better function in your macrophages.
Yeah.
You're going to have more M2 than M1, potentially. You're going to have neutrophils, which remove cell debris. They found out recently that they're crucial for nerve damage, because they remove the cell debris. They always assumed it was just macrophages, but it turns out that neutrophils do the lion's share of that work. And there's also some sort of level of redundancy where macrophages can do it too, but neutrophils primarily do it. And they remove the macrophages, and they found that the neutrophils did it regardless, and vice versa. It's crazy. There's a lot of redundancy in the immune system.
But neutrophil function. Go ahead, Vic.
You will really need to get a good study to see what is the microbiome and leaky gut, what part of inflammation is caused by senescent cells, what part is caused by just the whole
DNA damage.
The aging damage, because the body reacts itself and activates its inflammasomes. So it's a really hard mess to figure out, disentangle what parts.
Well, you guys all know my bias. The inflammasome can be activated by microbial products. Yep. So I always tie it back into this permeability, circulating microbial product story as a big driver of aging. But yeah, you guys know my bias in this. So.
And what about the ages, the advanced glycation end products that the body can?
There's some role. There's some data that methylglyoxal, which is a pretty potent age product, is antimicrobial. So age products may be a consequence of trying to fight the buggers off.
But you can also get lower the methylglyoxal levels due to your favorable microbiome, right? If you have a very good microbiome.
Yeah, and diet too. You can reduce it through diet. But then that goes back to the diet, microbiome, permeability, leak. The whole story.
I think it starts in the gut, as I said earlier. I think it begins in the gut. that's like the first sort of spark in the powder keg, and it literally comes out from there. Then that messes up the macrophages, which then causes problems with things like fat metabolism, clearance of senescent cells, clearance of cell debris, and around she goes again, and it keeps going around.
This is very interesting because they cited Eli Metchnikoff in the paper, immunologist, brilliant, back in 1908, and that was kind of a thing in the early 20th century when I used to read history, that people were obsessed with toxins building up in your gut, and that's what led to Kellogg's, right? It was the cereal that is laden with too much carbohydrates, but it was considered to be healthy by Dr. Kellogg, right? And it was promoted to give you a healthy gut, and people were so obsessed with it that they took it a little too far. I think there was a thing back in the turn of the last century where it was considered healthy to remove a big chunk of your intestine, so you wouldn't have toxins stewing in there.
It's ridiculous.
Yeah. Hopefully we don't go to that extreme, but there is a nugget of truth, right, in things that are, to some extent factually incorrect. But there was I recall reading an obsession about the gut and the toxins that are released early in the 20th century.
So it's ironic that Kellogg's would promote that was a gut healthy diet, but yet probably one of the primary components which can optimize your gut health, fiber, is generally missing from their products, right? But wait, Steve, just to take it back to what you're saying about starting in the gut, just to play devil's advocate, when you consider that germ-free mice are not immortal, there is cellular aging. And when you consider, right, there's cellular aging independent of the gut microbiome. So when you consider that the gut microbiome composition depends on host genotype, isn't it plausible to also speculate that because of cellular aging, independent of the gut microbiome, that the gut microbiome changes in concert with our cellular changes, that, just from the slow, whether it's oxidative stress or whatever it may be, you've got some minor amount of superoxide bouncing around, and slowly over time that slowly oxidizes us to death, right? And then the gut microbiome may change in concert to that, which then puts us into that even worse vicious cycle. So I think the root is fixing cellular aging and then being able to keep that youthful state.
Well, that ties in with hallmarks of aging, doesn't it? Because it puts inflammatory processes and what we call inflammaging as a secondary or antagonistic damage, which is consistent. Yeah. Because at the top, what you've got going on is DNA damage and mtDNA damage, which is part of genomic stability. Or instability, sorry, and it's consistent really, because it makes sense that the longest lived animals have the most stable genomes.
Yeah.
So I think what probably happens is as you say, genomic instability leads to changes in the gut, which then spark, and I think if we do fix those primary processes, we've got a strong case. That's why I'm very interested in the work of David Sinclair, who's working on NAD. A lot of people poo-poo it and say, oh, no, it's just resveratrol. But NAD is literally intrinsic to, so many cellular processes.
Sure. It's like ATP. It's a universal metabolite.
That's right.
But the issue I've got with his work, and I hope he's not watching or reviewing my grants, because I'm not trying to say all kinds of bad stuff, but if you want to say super physiological levels of TNF used in these papers, they are using 20-gram human equivalent based doses of NAD and nicotide, NAM, whatever it is, whatever that 20-gram human. So, and there's no data in humans using that much of a bolus. So whether or not it's going to work, I don't know.
But the interesting thing is also that inflammation, and CD38 in particular, is what destroys NAD in the first place.
But I don't think with the NAD studies, I mean, the lifespans don't exceed 900 days on average. Yeah. They increase from 800 to 800.
Yeah. Yep.
Guys, I'm going to have to bow out of this conversation because I have a meeting coming up.
Yeah, me too.
But you guys can feel free to keep talking. But I will have to exit the lab.
I think we've covered all the bases.
Excellent.
So that's great. Interesting to try something a little bit different. It's a little bit outside of our wheelhouse, as it were.
Well, that's something we don't really hear too much about, right? Aging and microbiome, it certainly wasn't in the hallmarks of aging. It's certainly a secondary response, right?
Well,
Is it secondary?
It is actually in hallmarks. It's kind of lumped in with the whole of the Inflammaging. They do talk about microbial.
But I mean, microbiome, not just Inflammaging, but the paper that I just pulled up, like the whole gamut of what the microbiome can offer in aging. Obviously for a metazoan, right? This is not going to apply for aging at the cellular level, but at the metazoan level.
It's interesting. So, maybe we'll explore NAD next time.
Maybe
it's the NAD world, fad world.
All right, stay tuned.
But thanks for everybody who's tuned in and watched it today. Hopefully it was as entertaining as ever, which is highly entertaining. And thanks very much to our heroes. Without you guys, we wouldn't be able to do this every month. And that's great. Thank you very much. We'll see you next time. Take care.